Good morning to everybody. Welcome to our webinar. I guess there's a few people joining us from Europe, so good afternoon to you all. Welcome to our webinar virtual forum where we're talking about when clinical benefit is clinically meaningful. My name is Russ Paulson. I'm the Chief Operating Officer at Us Against Alzheimer's. Since we were founded 12 years ago, we have been engaged in a relentless search to end Alzheimer's, which is, of course, the sixth leading killer in America. Our work centers on prevention, on early detection and diagnosis, and on equal access to treatments, regardless of race, gender, ethnicity, rural or urban. We give voice to patients, and we bring together collaborations from government, private sector scientists, academic scientists, and other organizations, all of whom put the us in Us Against Alzheimer's. Today, we are at a crossroads in Alzheimer's. It's been almost 20 years since drugs were approved to treat Alzheimer's, the core symptoms of Alzheimer's. And we now have two disease-modifying therapies with accelerated approval from the FDA. This is a huge accomplishment for the field and for the researchers, for the patients, for everyone who participated in that. Now, as decisions about these drugs are being made by regulators, by Medicare and other insurance companies, by hospitals, medical groups, ultimately by doctors and patients, there's kind of a basic question that needs to be answered. Is it a good drug for me? Is it a good drug that's going to work? Is the benefit meaningful to me? And for us, as a patient-oriented organization especially, it's important that that question be based, the answer to that question, it's important that the answer to that question be based on sound science, and importantly, what patients and caregivers think, the people living with this disease. What do they think? That's the question that brings us here today, and it's the question that Us Against Alzheimer's, through our Alzheimer's Disease Patient and Caregiver Engagement Initiative, or AD-PACE initiative, has been working on for the past almost 5 years. Our What Matters Most study has taken a scientifically rigorous approach to defining what matters to Alzheimer's. We're going to hear about that in the second half of today's panel. The researchers will present on really current research, stuff that's going to be published actually next week will be a big part of what they're going to be talking about. Before we get to the systematic scientific exploration of what matters most to patients, we want to get to the most urgent application of that science, the specific case, which is the results of the pivotal trial of Leqembi or lecanemab. So first we'll have those results, and then a discussion of what those results mean. Then there's been some discussion in the community about this term minimal clinically important difference, or MCID, in characterizing specifically Leqembi data. We spoke with the physician researcher who came up with that term, and we'll hear what he thinks, what he meant by that term, and what it doesn't mean. And then, as I said, we'll go to our panel reporting on the What Matters Most research, with discussants of patients, caregivers, and other Alzheimer's experts. Okay, so first, a presentation and discussion about the newest disease-modifying therapy for Alzheimer's disease, Leqembi. And we are pleased to welcome three experts in the field, starting with Dr. Michael Irizarry, who's the Senior VP of Clinical Research and Deputy Chief Clinical Officer at ASI. He's going to present the results of the trial that supported the accelerated approval. Dr. Irizarry leads clinical development for Alzheimer's treatments at ASI, worked in drug development in neurological and neurodegenerative diseases for years. He is a neurologist and has studied a long time the underlying biological mechanisms for Alzheimer's. And then after that, we will have a couple of discussants, and we'll have, I hope, a really important discussion. But first, the actual data. So Dr. Irizarry, take it away, please.
Thank you so much. If I could have the first slide up, please. So today I'll be presenting the lecanemab, also known as Leqembi, phase 3 results. Next slide, please. And then just a clarification from what Russ had mentioned. On January 6th, lecanemab was approved for the treatment of Alzheimer's disease under the accelerated approval pathway. And that was based on the phase 2 data and the reduction of amyloid plaques in those treated patients. And that particular approval was for mild cognitive impairment due to AD or mild AD dementia with confirmed elevated amyloid, the population that was studied in clinical trials. So continued approval for this indication may be contingent upon verification of the clinical benefit in a confirmatory trial, and Clarity AD is serving as that confirmatory trial. So the accelerated approval is based on the phase 2 data, and we have submitted the phase 3 data for the full traditional approval. So today I'll be presenting the results of the Clarity AD study, the phase 3 investigational confirmatory trial of lecanemab. And this was submitted on January 6th as a supplemental biological license application to the FDA to support the conversion of the accelerated approval to a traditional approval. And that BLA is subject to validation of the acceptance of the application for review. So with that, we can move on to the next slide. Lecanemab is a monoclonal antibody that preferentially binds to soluble aggregated amyloid beta protein. Amyloid beta protein is the component of amyloid plaques in Alzheimer's disease that begins to accumulate years before the onset of symptoms. And lecanemab binds to these soluble aggregated species known as oligomers or protofibrils, about 10-fold higher affinity than to amyloid fibrils that form the plaques. So lecanemab clears these soluble aggregated species and also clears amyloid plaques through microglia clearance processes. Next slide, please. The clinical trial that I'll be talking about is called Clarity AD. It was a global phase 3 study, placebo-controlled, double-blind, parallel group, randomized study that enrolled 1,795 patients with early Alzheimer's disease, meaning mild cognitive impairment due to Alzheimer's disease or mild AD dementia with confirmed elevated amyloid in the brain. Inclusion criteria required a mini mental state score between 22 to 30. And patients also had to demonstrate impairment of recall based on a neuropsychological test, the Wechsler memory scale for logical memory. So patients were randomized 1-to-1 to either lecanemab, 10 milligrams per kilogram IV, administered every 2 weeks, or placebo for 18 months of treatment, after which people could roll into an open label extension of lecanemab. The primary outcome measure for the clinical trial was the clinical dementia rating scale sum of boxes, the change score from baseline at 18 months. And that scale is an 18-point scale that is a patient and caregiver interview that assesses 6 domains of cognition and of function that are of importance for patients and caregivers. The key secondary outcome measures were amyloid clearance by amyloid PET scan, a cognitive test administered to the patient, the ADAS-Cog14. The ADCOMS was a composite scale that we had used in the phase 2 study that we included to be able to compare to the phase 2 results. And then a scale of daily functioning, the ADCS-MCI, Activities of Daily Living scale. There was also extensive biomarker studies that assessed the impact of treatment on the amyloid pathway, the tau pathway, the neurodegeneration pathway in Alzheimer's disease. And I won't discuss those results in this presentation. Next slide, please. So first, I'll go through the primary and key secondary outcomes. In these graphs, the change from baseline is on the y-axis, time is on the x-axis. The lecanemab treatment group is in green, and the placebo treatment group is in black. In the CDR sum of boxes, there was at 18 months a 27% slowing of decline by lecanemab, with an absolute difference at 18 months of 0.45 on the scale. And then we saw that over time, the difference between the treated group and placebo, the absolute difference, increased over time. When we looked at the domains of the CDR scale, memory, orientation, judgment, problem solving, community affairs, home and hobbies, and personal care, the effect was seen across measures of across all the domains, both measures of cognition and of function. Just to give more background on the scale, I mentioned that the scale is an 18-point scale. But we're really looking at the very front part of the scale. So the mean baseline score on CDR sum of boxes was about 3. And you can see that the placebo progression was 1.6 over 18 months. So the range of the scale that we're actually looking for a treatment effect in is between 0 and 1.6. And so when we consider in that framework, we see a 27% slowing of progression on the CDR sum of boxes. Next slide, please. Lecanemab reduced amyloid in the brain as early as 3 months, and by 18 months, about 68% of the people were under the quantitative threshold for amyloid positivity. Essentially had become amyloid negative based on quantitative criteria. Next slide, please. The ADAS-Cog14 is the cognitive scale, and there was 26% slowing of decline on cognition at 18 months by lecanemab. We saw separation as early as 3 months, with increasing separation over time, except just for that last 3-month time point where the lines look parallel. Next slide, please. The activities of daily living scale is a measure of functioning, and lecanemab delayed progression or delayed loss of functioning by 37% relative to placebo, with differences seen as early as 6 months and the absolute differences increasing over time. Next slide, please. So the CDR sum of boxes, while it's the gold standard for clinical trials, is not routinely used in clinical practice. So we did a couple other analyses to try to understand what these effects that we were seeing on the CDR sum of boxes and ADAS-Cog or the ADL scale, how they could translate into concepts that could be explained to clinicians or patients who don't use the CDR sum of boxes. So this particular analysis looked at time to progressing to the next stage of Alzheimer's disease. So what proportion of people progressed from essentially mild cognitive impairment to mild AD dementia, or from mild AD dementia to moderate AD? And you can see that in this graph, the people on placebo are progressing to the next stage of disease at a faster rate than people that were treated on lecanemab. So the hazard ratio, or the ratio of the incidence rates for lecanemab versus placebo is 0.69, meaning that the rate of progressing to the next stage of disease is 31% lower in people treated with lecanemab, or alternatively, individuals remain in earlier stages of Alzheimer's disease for a longer period of time. And we could detect that even within this 18-month clinical trial. Next slide, please. The other approach was to try to translate the amount of the treatment difference in CDR sum of boxes to a time scale, so to a time preserved. And one way to look at it is to add a best fitting line to the CDR sum of boxes for the lecanemab treatment group in green and the placebo treated group in black. If we follow the green line all the way to 18 months, you can see that the lecanemab treated group had declined by 1.16 on the CDR sum of boxes. However, the placebo group has already reached that point approximately 5.3 months before. Lecanemab has kind of preserved those 5.3 months of time. And if we project out, these lines separate all the over time, and the amount of time preserved can potentially increase. Next slide, please. We also look to see how these effects translated into scales of health-related quality of life or caregiver burden. Up on the top, we have two quality of life scales that are administered to patients themselves, and the subjects themselves rated approximately 50% slower decline in their health-related quality of life. When the caregiver assessed the quality of the life of the patient, there was a 23% less decline based on the caregiver report by proxy. And then finally, the Zaret burden interview assesses the burdens and challenges of caring for a patient with dementia. And we saw a 38% slower decline in the burden of caring for a patient with lecanemab treatment. Next slide, please. Now, I know we're talking about clinical meaningfulness, but it's important to understand the clinical meaningfulness in the context of the safety and the benefit risk of treatment. So I'll spend a few slides highlighting the important adverse events that can be seen with lecanemab treatment. Within the double-blind phase of the study, there were a similar number of deaths between the lecanemab and the placebo-treated group. In terms of serious adverse events, these are adverse events that may require a hospitalization or could be life-threatening. The rate was higher in the lecanemab group versus the placebo group, 14% versus 11.3%. And these were serious adverse events associated with amyloid-related imaging abnormalities, a class of abnormalities that are seen with amyloid-lowering drugs. Either ARIA-E, which is related to increased fluid in the brain or edema, or ARIA-H, which can show up as small areas of bleeding in the brain, microhemorrhages or siderosis, or in rare cases, larger intracerebral hemorrhage. So the rates of SAE for ARIA-E and ARIA-H were 0.8 and 0.6% respectively. There's also a higher rate of infusion-related reaction SAEs associated with lecanemab. Next slide, please. In terms of the most common adverse events for lecanemab versus placebo, the most common adverse event was infusion-related reaction. 26% of people who were treated with lecanemab versus 7.4% treated with placebo. These tended to occur with the first dose of treatment and could be manageable with symptomatic medication. The rate of amyloid-related imaging abnormalities edema was 12.6% for the lecanemab group and 1.7% in the placebo group. And the rate of ARIA-H was 17.3% in the lecanemab group versus 9% in the placebo group. Much of the excess ARIA-H was ARIA-H that tend to occur at the same time as ARIA-E early in the course of treatment. Next slide, please. With regards to ARIA-E, ARIA-E events were largely mild to moderate radiographically and asymptomatic. They tended to occur within the first 3 to 6 months of treatment and tended to resolve after about 4 months of treatment. The rate of ARIA-E increased with the number of ApoE4 alleles that a person carried. And while the overall rate of ARIA-E was 12.7%, the overall rate of people that had ARIA-E that had symptoms was 2.8%. But again, the people that had 2 copies of the ApoE4 allele were those at highest risk for ARIA and for symptoms of ARIA. And symptoms of ARIA commonly included headache, visual disturbance, or confusion, although in severe cases, it can include focal neurological findings and seizure. Next slide, please. The rate of ARIA-H is broken down in this slide in terms of the rate of microhemorrhage and superficial siderosis, which is increased in the lecanemab group relative to placebo, but generally does not have symptoms. Lecanemab treatment did have a higher number of people with cerebral macrohemorrhages. This is bleeding in the brain that's greater than 1 centimeter in size. So as part of the treatment with lecanemab, assessment of the potential risk for ARIA before treatment is recommended in the product insert, including a baseline MRI to assess for cerebral amyloid angiopathy, consideration of ApoE4 testing to identify ApoE4 homozygotes, and discussing the risks of anticoagulation treatment with lecanemab. Next slide, please. So in conclusion, there was a convergence of evidence of lecanemab treatment effects across multiple measures of cognition, function, disease progression, health-related quality of life, and caregiver burden. In addition, there was an impact on multiple amyloid tau and neurodegeneration biomarkers that supported target engagement and biological disease modification. And the safety profile includes important adverse events of infusion-related reaction, ARIA-E and ARIA-H. Thank you.
Thank you, Michael. Joining us now to discuss what Michael just went over, which could have taken 45 minutes probably, and thank you for doing it so quickly, Michael. Joining us are 2 clinician researchers, Dr. Monica Parker, Assistant Professor of Medicine at Emory University, family medicine physician by training, who regularly treats Alzheimer's patients as a geriatric primary care provider, also the lead for the Minority Engagement Core at Emory's Alzheimer's Disease Research Center, and Dr. Jeff Cummings, research professor at the Department of Brain Health at the University of Nevada, Las Vegas, and a widely published author on Alzheimer's research. If you look up the most published authors in Alzheimer's, you're going to find Dr. Cummings' name right at the very top. So thank you for both for joining us. I want to start with you, Dr. Cummings. The phase 3 trial results that Dr. Irizarry just presented showed statistically significant benefit versus placebo in a lot of different tools that measure sort of symptoms, and also in the clinical pathology, the brain plaques and that kind of stuff. When you see that kind of concordance among the various measures, what does that tell you?
Thanks, Russ, and thanks, Dr. Irizarry, for a great presentation. These data are so exciting to me. It was 2003 since we last had a drug approved before aducanumab in 2021. And these are drugs that affect the underlying biology of Alzheimer's disease. This is a whole new way of thinking about therapy for Alzheimer's disease. So I am really excited about this. And the convergence of data, Russ, that you mentioned is very important in terms of our ability to embrace this data as truly validly reflecting the effect on the disease. So the CDR sum of boxes, for example, that is mostly done by a clinician interviewing the patient and asking the patient directly, do you remember this? Do you remember that? Then, of course, there's the caregiver measures, and the caregiver measures also showed a drug placebo difference. And there's the biological measures that showed these very dramatic drug placebo differences. There was also a convergence in terms of function, in terms of cognition, in terms of quality of life. So all sorts of dimensions of the patient's life have come together and reflected very similarly. Now, the magnitudes were different, right? It was 27% on the CDR sum of boxes. That's very important. That's the primary. But it was 37%, almost 40% on the ADL function. And the ADL function is so important to patients. This is, what can I do today? What can I do tomorrow? These are the things that are so important to patients. So the convergence of data, really, really important in terms of showing us the breadth of effect of treatment.
Thanks. And so really positive news on that regard. Dr. Parker, a lot of your career focus, or your current job focuses on recruiting minorities and improving minority recruitment. And the results on that for this particular pivotal trial were not altogether successful. 22.5% Latino engagement, but only about 4.5% African Americans. And Latinos and African Americans are disproportionately affected by the disease. So how do you think about that in this context and in terms of the patients that you'll be serving?
I think that what's clear is that people in these communities are at a disadvantage because, number one, they're not represented in clinical trials, mainly because there's probably not been an access point for them. And having an access point for clinical trial participation means, for the most part, that you have a medical doctor, whether it's your primary care provider who refers you to a neurologist who is associated with a research entity or research center. Here at Emory University, I'm a part of one of 33 nationwide research centers funded by the ARIA-H that allow this kind of research to take place. Other than that, there are for-profit centers that conduct clinical research. So the important piece, and this is something that's been highlighted certainly in the pandemic, is that certain populations have greater clinical access, first of all, to a qualified medical provider who can make an accurate diagnosis about whether or not you have Alzheimer's or some other type of dementia. Because I do educational outreach and I'm in the community, I know that the general population does not know the difference between Alzheimer's dementia or any other kind of dementia. As far as they're concerned, it's memory loss or some kind of functional status loss. What they don't always understand is that that can be caused by a number of different things. And it's really important to understand what those causes are, such that you can more specifically direct treatment to whatever the cause is. And I think that this trial, much like any others, has not enough minority representation. And I think that the main reason that occurs is because these patients, and I will say patients, just are not affiliated with medical providers who are associated with a research entity that conducts this kind of research. So if you want more participation, you're going to have to make it more likely that people who are affected are being served by providers who are aware and can make access to research trials possible. And you don't do that by putting a flyer up on a bus stop or a transit stop.
Thanks. Lots of room for improvement there, I think. Kudos, Dr. Irizarry, to ASI for doing such a good job with the Latino community, but obviously we all have some work to do in general. I wanted to ask you, oh, sorry, go ahead, Michael.
Yeah, and I completely agree with Dr. Parker's statements. We did initiate a range of activities to try to improve the access and the representativeness in the clinical trial. The key activities we did was early on try to select investigators that had access to diverse communities. Also tried to build trust in these communities through outreach, either to community centers or faith-based groups. And I think also accelerated by the pandemic, trying to initiate more decentralized clinical trial activities to reduce the burden of participation in clinical trials, which can be a challenge in certain for people, especially with a drug that's administered every 2 weeks. I agree there is more work to be done. We in the AHEAD 345 clinical trial, we are looking at other approaches as well to try to improve access and representativeness to these clinical trials.
Russ, can I ask a
Yeah.
question of Dr. Irizarry? One of the things that's been bothering me is that if we achieve representative populations in our trials, those representative populations will be too small to give a statistically valid assessment of the outcome. So how do we manage that? Should we have separate trials for different ethnic groups to really understand efficacy and safety? Or should we simply try to get enough patients in the trial in order to draw statistically significant conclusions? How can we make the participation of minority members more meaningful in terms of understanding the effects?
So in terms of especially a phase 3 clinical trial where we would optimally want to understand the safety and efficacy in a population that's reflective of the disease and that would be eligible for treatment. I think it's as important as possible to try to represent that range of people that would be eligible for treatment. And to it's very difficult to do a separate clinical trial for each group that you could think of. But there is an analysis in the clinical trials that can be done to see whether there's any evidence of significant heterogeneity that could be followed up. So we published in the New England Journal, for instance, a whole range of subgroup analyses that can then serve as a basis for further study.
Thank you.
Thanks. Dr. Irizarry, I want to stay with you for a second on another sort of topic that you aired pretty clearly during your presentation, which is the risk side. So we're mostly talking about the benefits here, but benefits matter really a lot in the context of risk. So, and then there's been some press statements about some risks and dramatic cases in a couple of cases. But even if you look at the pure data, there's risk here. So as a neurologist, how do you think about it? And as an ASI employee, what's ASI doing about it?
So in terms of the benefit risk discussion, really needs to be focused on the label, but needs to be an individualized discussion between the physician and the patient with an understanding of a balanced discussion of the potential benefits and the potential risks, and also identifying in particular the patients that might be at highest risk and where that discussion will have to be more data focused and also understanding the preferences of the patient and the clinician. So being able to discuss ApoE genotyping, being able to discuss what the baseline MRI shows and that risk that implies and their comorbidities. And so it is a more nuanced discussion. ASI has initiated an education initiative on amyloid-related imaging abnormalities, understanding ARIA.com, which is more general in terms of understanding the incidence, the kind of staging, understanding the severity. So the education will be an important component. And then also the ALSNet registry, we are encouraging physicians to participate in, and patients to participate in. as another approach to continue to gather information on amyloid treatments as they become available in the market.
We're in a new era, right? I mean, doctors aren't used to prescribing this stuff, and having conversations and understanding how to monitor safety, I think, is going to be a big change for the field and change for practice. Dr. Cummings, I wanted to ask you, so we talked earlier, and you sort of in your opening comments talked about we're in a new era. And the current era is this specific sort of monoclonal antibodies targeting amyloid in the brain. And that sounds like a thing, but there's multiple drugs within that category. And Dr. Irizarry's presentation talked about sort of different species of amyloid beta that are targeted. But how do we think about the differences? There are similarities, obviously. They're all targeting amyloid of some sort, but how do you think about the differences among the different drugs that are either on the market or coming to the market in this category?
A few thoughts about this, and of course, Dr. Parker and Dr. Irizarry have ideas as well. First of all, you mentioned that there are different types of amyloid in the brain. So it's first produced as a very simple molecule, becomes much more complex as the different molecules begin to stick together, and finally they form a plaque. And the different antibodies then target different species within that whole range of amyloid. So one big difference among them is the target, the specific target of the amyloid that they have. Another, of course, is infusion versus subcutaneous administration. We know that the gantenerumab program did not achieve a statistically significant benefit and was administered subcutaneously, but it may well be possible to administer drugs subcutaneously and achieve a clinical benefit. So differences in administration are likely to be observed as well. I'd also like to stress that we're very early in this process. And in truth, you can only compare molecules if they're in the same trial. That is because therefore randomization allows you to know that the patients were more or less equal in terms of their exposure. So we'll probably never have quite the fine-grained data that we would really like to have for the different monoclonal antibodies, but we do see that there are differences, and we anticipate those differences playing themselves out in terms of care choices for patients and by patients.
Dr. Irizarry, anything you wanted to add?
I think the other feature that we've seen with antibodies that clear amyloid is the relationship between the speed and the amount of amyloid reduction and the clinical efficacy that led to the amyloid being a reasonably likely surrogate for the FDA for accelerated approval. And across these monoclonal antibodies, there does seem to be a relationship between the amount of amyloid clearance and the amount of clinical efficacy that can be expected. I'll also, in addition to amyloid, the A beta species, there's still progression in people even as they're treated with monoclonal antibodies. The progression may be slower, but the disease is still progressing, highlighting the opportunities for other targets within the Alzheimer's cascade, targeting tau or targeting neurodegeneration. And also the rationale for taking monoclonal antibodies earlier in the course of disease in preclinical AD and testing them in that population before there's been significant irreversible neurodegeneration.
Thanks. And Dr. Parker, just switching to you as someone who sees patients regularly, what more do you need to hear or learn about before you feel sort of comfortable that this is something you want to incorporate in your regular practice and shared decision-making with patients?
I think that as a primary care clinician, I don't think that I personally have enough information to advocate for or against. One of the things that would be a negative or against would be, number one, we don't really know how these medications are going to be paid for. And that's a real strong consideration for any provider. The average patient is not going to be able to apply to afford this medication without significant insurance coverage. And right now, that's still a question. Even with the Aduhelm, which has been FDA approved, there's still there is coverage, but lecanemab doesn't have that as of this moment, unless something's happened in the last 24 hours. So the other things that we've talked about earlier, managing adverse events, hemorrhages, that adds to the burden of cost, if you will. It's an infusion. It's like anything else. There's cost that's related to that. And I think that for any clinician trying to advise his or her patient, cost is going to be a major factor for whether or not I can administer it in my office or my outpatient setting. Because it's infusion related, I'm in an academic institution or affiliated with SAME. It's likely going to be a hospital administered drug, and those are the things that need to go into that discussion. So I think cost, availability, and management of those things that we've identified that may be adverse kinds of effects are important for patients to understand, but for the average clinician, whether it's a primary care clinician or a neurologic clinician, how am I going to manage those other things that are affiliated with this?
Thanks. Dr. Irizarry, I think part of what starts to make the access wider under the current paradigm from CMS, at least, is triggering traditional approval. And you talked a little bit about that early in your discussion, but I wonder if you can talk about that and potentially other things that Dr. Parker brought up.
Certainly. I can talk a little bit about the Clarity AD data and the lecanemab clinical development data and the CMS NCD. We've had very productive interactions with CMS where we've gone through, even before Clarity read out, we went through the data and the type of results that we were hoping to generate. And then after the Clarity AD results, had discussions with them in terms of how the results address their three key questions as part of their coverage with evidence determination. So the clinical meaningfulness of the benefit, the impact across different subgroups, the benefits and harms over time. So our hope is that the data generated can address those questions and support reimbursement. So the approach is really to be very data driven. As well as understanding that there will continue to be data generated based on ongoing trials and the existing voluntary registries such as ALSNet.
Thank you.
Russ,
So,
maybe I could address
yeah,
one
yeah.
thing that Dr. Parker brought up that I'm particularly concerned about, which is these are complicated drugs and they're brand new drugs. They're unprecedented. It's great to have them, but we have to use them well, and we have to learn how to use them well. So we have developed a group, the Therapeutics Working Group, that develops appropriate use recommendations. They're not guidelines or criteria. They are people, experts in the use of these drugs who are trying to provide clinicians some guidance and to link the prescribing information that the FDA provides and the guidance from the trial to what practice actually looks like. And I think these are the kind of gaps that new exciting therapies create, and we have to find innovative ways of responding to those gaps.
Thank you. So we're closing in on the end, but I wanted to ask one important question. Dr. Cummings, I mentioned you've been published a lot. You've been working on Alzheimer's for a couple of years now. And we're about to launch into a discussion about what matters most to patients. And this whole thing is about clinical meaningfulness and whether something is clinically meaningful and risk and benefit. And it would all be a moot point if we could just cure this disease. If we had the penicillin for Alzheimer's, we wouldn't be having this discussion at all. What is it about Alzheimer's that keeps that from happening? Why don't we have a cure?
I'll take that on. I'm sure other people have views as well. First of all, I'll put it in the context of chronic disease, which is we can cure infections, but we don't cure heart disease or lung disease or liver disease for the most part. If it's an infectious liver disease, we can. So chronic diseases that occur more in older people are managed. They're not cured. And it's true across the board of the organ systems, including the brain. In the case of Alzheimer's disease, it appears that there is a group of people who clear amyloid less effectively from the brain during their lifespan. And those who do so to a critical level, that is, they are not able to rid the brain to a critical level, they will get Alzheimer's disease. And that's exaggerated by certain genetics. The ApoE4 gene makes it more difficult to clear the amyloid from the brain. So this is intimately tied in with aging. It's not aging, it's abnormal, but it is tied in with changes that occur in the course of aging. And so I think a cure probably isn't the right conceptual framework for this disease. I think we're trying to, here's my goal, keep my patients functioning at the highest level for the longest period of time. That's my goal. It's not to seek a cure, which I think is not likely given what we understand about the disease.
Dr. Parker?
Well, I'm in full agreement. I think that when patients are talking to their doctors, you have to guide the discussion and kind of set the stage for appropriate expectations. It's like, okay, you can get this medication, but you need to understand that this is not a medication that is a cure, a definitive cure. And it's something that slows down the process. And I think people understand that to some degree, but they want something visible and they want something that makes sense to them. So when Dr. Cummings says, I want you to be able to function for as long as you can, as independently as possible, I think that that's what most people would like. I think that it's, I agree with Dr. Cummings. I think that you need to set the expectation that there's no cure, but we're going to get to the end, but we just may not get there as fast, and be clear about what those objectives are. And so maybe it's being able to feed yourself, the basic things that burden caregivers, feeding, grooming, toileting, all those sorts of things. As long as somebody's able to do that, it's not a problem. We don't mind confusion, but we really have a hard time when we have to assist somebody that we love and we live with those things on a regular basis, because that's burdensome. So as long as I can be independent in that respect for as long as I can be, I think that's a win for patients.
Thank you. And finally, Dr. Irizarry, I'm sure if you could have come up with a cure, you would have been happy to do so. But your final word on this.
Yeah, I think scientifically it's important to recognize that these late onset neurodegenerative diseases have been particularly intractable to disease modifying treatments. So there's symptomatic treatments for Parkinson's or for Huntington's, but these diseases have been pretty relentless. And the hope is that these data from amyloid lowering antibodies do indeed support that these diseases can be treated, that there can be approaches that can be tested to slow down progression, that hopefully will be generalizable in terms of some of the concepts that we're talking about in terms of clinical meaningfulness and progression to other neurodegenerative diseases as well.
Thank you, Dr. Michael Irizarry, Dr. Jeff Cummings, Dr. Monica Parker. for a almost hour that whizzed by on a really important topic. And I really appreciate all of you making time for this. Up next, we'll be hearing from the guy that invented the term minimal clinically important difference, Dr. Roman Yeshka at McMaster University, then a panel on what matters most to Alzheimer's patients as we've discovered it through some pretty rigorous scientific research. So first, Dr. Yeshka.
Hi, everybody. I'm John Summers with Us Against Alzheimer's, and today I'm joined by Dr. Roman Yeshka. He's a highly respected researcher, clinician, and professor at McMaster University. Dr. Yeshka, thank you so much for joining us today. Really appreciate having you.
Oh, my pleasure.
So let's jump right into it, if you don't mind. In 1989, you wrote an article that introduced the concept of minimally clinically important difference, or what we often refer to as MCID. And I want to read what you wrote then and then get a little bit of a better understanding from you. You said MCID is the smallest difference in score and the domain of interest which patients perceive as beneficial and which would mandate, in the absence of troublesome side effects and excessive cost, a change in the patient's management. What does that mean in simple terms?
The question here is how much you would need to improve your health status in whatever way you defined it, physical, psychological, functional, in order to go through the exercise of subjecting yourself to the regimen, which may have, again, different degree of effort, cost, time requirements, social function requirements, and so forth.
And when you're talking about benefit, you're talking about in patients' daily lives, right? What they perceive as benefit, right?
The evolution of this concept is reflected in names. We called it minimal clinically important difference, and somehow clinically was associated with profession, with clinicians. We actually moved from minimal clinically important difference, MCID, to minimal important difference, because as we were applying this concept, it became clear that the judge and jury about what is important should not likely be a clinician, but should likely be the most interested party in this equation, which is the patient. What is minimally clinically important to a clinician may be much less important to the patient and may be totally unimportant to a caregiver. So the importance of defining who is the judge and jury in terms of what is important is absolutely crucial.
Would a scale that measures whether a clinician believes that there has been, in their view, significant difference since the last time they saw a patient be a good way to measure MCID?
The clinician assessment and clinician judgment is an important one, but I wouldn't say also that it is the most important one or the only one. So I wouldn't dismiss it offhand, for example, saying, no, we are only interested in perceptions of the patients. I would be interested in perception of clinicians, especially at the stage of a generation of such instrument for the patients. Because if those instruments measuring the physician impression or clinician impression in general, and patient's impression and family impressions, if they are discordant, we obviously have a problem which we have to resolve.
Yeah, and you have really come up with a lot of terms to help people get a better understanding of this. And in fact, since 1989, you introduced 2 additional terms to your work, including patient important outcomes and human important outcomes. Can you tell us why that was important to you to do that? And then also tell us the difference between the 2?
What became very clear is that if we tell the patients we would like to treat you because your hemoglobin A1C will drop by 0.5%, despite all the advertisement in TV, people have no clue what it means. And obviously people have no idea what it does to their well-being or function. So around the beginning of 1990s, again, we came, instead of talking about important changes, we started to talk about patients' important changes and patients' important outcomes. And that required that rather than asking what's your FEV1 or what is your hemoglobin A1C, we need to ask the patient, how are you feeling today? And do you feel any better since we last seen you, or do you feel any worse, or do you feel unchanged? But equally important people are around, people who may be dramatically affected by therapeutic or administrative decisions which we are making. And here we came with the label of human important outcomes. It's not only patient which counts.
What do you say to those who question the value of the patient experience?
It would be extremely patronizing way of approaching therapeutic process. I do not believe people would have this, again, extreme approach of devaluing patients' experiences. I think there may be people who value, again, physiological parameters and what we can measure through our measurement instruments. There is a value in both approaches. It almost looks to me like ideological or philosophical debate here. The goal of our profession is to influence how people feel and how well they live. And the ultimate judges here have to be those who are subjected to it. We may think whatever we want, but in the end, our patients and their close by simply know better.
Well, Dr. Yashka, thank you so much for the important work you do, for your insights this morning, and for being an important part of this important conversation we're having today. Thank you so much.
Thank you for having me. Goodbye.